A recent study published in Nature Aging showed that age probably doesn't progress in a straight line, and instead, aging might be curved or change speed at different times in life. Now that's a remarkable finding, but what's equally remarkable is that the scientists who made these discoveries used not just 1 or 10 or 100 different laboratory measurements, but instead 135,000 different laboratory measurements. We'll talk about what they learned, what this might mean for aging science, and a new branch of science that's driving these kinds of discoveries, a fusion of computer science and biology called multiomics. One of the biggest challenges preventing us from understanding the root causes of aging is that there's been too much information. Over the last century, medicine's been focused on linking individual diseases with one or a handful of laboratory results. And those lab tests are typically the ones that we know because we learned them in medical school. But take a look at this video and imagine trying to predict the flight patterns of this entire flock of birds by tracking just one or a handful of them. If we zoom out, the reality is that there are many hundreds or thousands of flight patterns that we could be tracking, just like there are now many thousands of molecules that we could be measuring in each person. So the question then becomes, how do we take this massive amount of data that we can acquire per person and distill it down in a way that doctors and scientists can understand? Well, one answer is that we could use mathematical techniques to collapse down a large number of variables or dimensions into just a few easy to understand summary measurements. And that's what the researchers did in this 2023 study, taking data from 108 subjects and showing that these summary measurements correlated with age. They did this using a technique called principal components analysis. And because this technique is so common in the literature, I want to take a moment to walk through what that looks like. A simple illustration is to imagine that you're running a 5K on the Appalachian Trail. So from the sidelines, people are trying to help you, but they're yelling out the latitude and the longitude and the altitude of your competitors. But that's too much to think about. A simpler way to represent where the other runners are is to take the axis that represents the greatest variance in all of the runners and collapse all those variables down onto that one axis. That axis just so happens to be the exact path of the trail itself. So now you start to hear from the sidelines that there's one runner a quarter of a mile ahead of you, and there's one runner a quarter of a mile behind you. So that's an illustration of a prototype of how many of these dimensional reduction algorithms work in these kinds of large studies, and you'll see them more and more in the literature. But no surprise here, the summary stats correlated with age. We see lots of things correlate with age. What we really want to know is whether aging progresses in a straight line or whether it's curved and speeds up and ebbs and flows. We can start to see the answer to that question here. The first thing you might notice is that none of these clusters progress with age in a straight line. But let's talk about what we're actually looking at. They use a technique called C-means clustering, where every data point is assigned a probability score of belonging to a particular cluster. So the researchers here identified 11 different clusters and highlighted these 3. The scores in the clusters on the left and on the right, clusters 2 and 5, tended to stay relatively steady until about age 60, where they then subsequently took off. And in those clusters, the researchers found markers that tend to be involved in pathways like DNA structure, programmed cell death, mRNA stability, autophagy, and more clinically speaking, kidney function and diabetes risk, which of course is a huge risk factor for aging. For the cluster that you see in the middle, the opposite was true, staying relatively steady and then dropping off after 60. Those pathways tended to be involved in oxidative stress and fatty acid metabolism. So we're getting a strong sense now that aging may progress in a nonlinear or a curved fashion. But the next logical question would be, at what ages do we see the biggest differences? To be able to answer that question, the scientists use something called a sliding window technique. And they ask the question, to what extent do we see differences from baseline across the span of aging? The results here are striking, and they've been getting a lot of press. So the researchers identified 2 peaks, ages 44 and 60. What makes these 2 peaks interesting is that they don't just show up in the overall summary analysis, but across a wide range of measurement types, including protein levels, mRNA transcripts, which are the blueprints for our cells to be able to make the proteins that we need, metabolites, cytokines, and microbiome measurements. And many of these pathways had overlap in both of these peaks and tended to be related to aging. So is this an open and shut case that aging accelerates at certain ages? Let's take a step back and see what these data are telling us and what they're not telling us. First, aging probably doesn't progress in a straight line. Second, whatever's happening in the mid-40s and around age 60 is happening across multiple different systems. Whether these changes are physiologic, pathologic, or compensatory, the body trying to be able to deal with its disease, remains to be determined. And third, all of the measurements in this study were explored with regard to chronological age. This is not a measurement of health or disease. What many are interested in is healthy versus unhealthy aging. And then lastly, it's been shown that different cells and different organs progress differently across aging. It wasn't possible in this study to obtain data on what's going on in, for example, a liver cell or a heart cell. So that remains to be determined, and it's likely just scratching the surface, albeit a very solid start to be able to understand what happens across the lifespan. Thanks for listening. If you want to support this channel, the very best way to do that is to hit the like and subscribe buttons. If you're on Spotify and are able to head over and leave a review, it helps so that the algorithm can pick up the channel and help it reach more people. I'll link to the Spotify channel in the description. With that, thank you again, and we'll see you next time.